There we go. Thank you so much. Helen, it's all yours.
Okay, great. Thank you. Sorry, my internet was a little bit unstable there, so I want to make sure we do this right. Can you see my, oh, just one moment. There, can you see my slides?
Yes.
Okay. So I just, hopefully you can hear me. It looks like my internet's a little bit unstable. Just can you nod if you can hear me?
Yes, we can.
Okay, perfect. Thank you. Sorry about that. So I want to step back a little bit and really share, the next step from genomics to transcriptomics, and get into a little bit out of the chronic conditions and more, out of the rare conditions and into some of the common conditions. One of the biggest problems that's facing the world today is that of chronic disease, right? More than 50% of the world lives with some kind of chronic disease, and it accounts for 60% of deaths worldwide, and the incidence just keeps increasing. Now, most of us know that diet has a big impact on the incidence and the outcome of many of these chronic diseases, but there's a lot of confusion as to what the right diet is that's going to prevent it. We've all heard of ketogenic and paleo and Mediterranean and the USDA food pyramid, and much of the popular press is really just full of contradictions. Which diet is right for that individual? So part of the problem is that up to now, we've really been looking at the tip of the iceberg. We're using epidemiological and population studies, but we really need to look underneath the surface at each person's molecular uniqueness to really understand what diet is right for each person. So I just want to take a brief moment and take a look at underneath that surface. But first of all, I want to remind everyone that we're not really human, right? We're a complex ecosystem of human and microbial cells and genes that are all interacting with each other in our environment, especially our diet. And this was the cover of The Economist magazine a number of years ago, that microbes maketh man, and it's becoming more and more true as we learn more and more. So I think everybody understands the microbiome. Every surface inside and out of our body is covered by microbes, with the largest collection of them being in our gut, over 100 trillion organisms, and they're at least equal to the number of human cells. And when we look at a genomic perspective, we can see that we're outnumbered by microbial genes about 150 to 1. So these microbes do so many things for us, and these are just a few of them, just a partial list of the functions that they perform. And one of the most essential things that they do is orchestrate our immune system. So 70% of our immune cells live just on the other side of our gut wall. And there's a very intimate interaction between these microbes and our immune system, so much so that they actually train it and control our immune system. So this is one of my favorite quotes. The mammalian immune system, which seems to be designed to control microorganisms, is in fact controlled by microorganisms. So when you think of all of these things, it's no wonder that there's not one chronic disease that you can think of that's not been associated with a disruption of our microbiome, at least at some level. And the microbiomes not only influence our health, as we've talked about, they influence so many other things, including our response to drugs. A new term, rather than pharmacogenomics, we're talking about pharmacobiomics, is the impact that the microbiome has on our response to drugs. They control our biological clock and our mood. And of course, there's the gut-brain connection that we're all aware of. So this is a quote from Martin Blaser. It's reasonable to propose that the composition of the microbiome and its activities are involved in most, if not all, of the biological processes that constitute human health and disease. So what makes a healthy microbiome, and how can we tell if it's been disturbed, and then what can we do about it? Well, we know that one of the most important factors that promotes a healthy microbiome, of course, is our diet. It's one of the main reasons that diet has such a big effect on chronic disease is because it impacts our microbiome so strongly. So the question that you need to ask, of course, is what's a healthy diet? And like we alluded to at the beginning, the answer is there is no one healthy diet that's right for everyone. Since we all have such unique microbiomes as unique as our fingerprints, our optimal diets will also be unique. So we understand, and I think there's a general consensus out there that the microbiome is very critical and important to our overall health. But the big criticism has always been, yes, we understand that, but how do you know what to do about it? And the answer is we've just not been using the right tools up until now. You know, when we started, our tools were very limited. We used culture to try to grow the organisms in our gut. Now, of course, 80% of the organisms are anaerobic. So as soon as they're exposed to oxygen, they're going to die. So we miss about 80% of what's there using this technique. And we can't identify eukaryotes or viruses at all. 16S, ribosomal RNA gene sequencing, is another step in trying to understand what's going on in the microbiome. This is what's commonly being used by most researchers and companies today, and it's looking at a small piece of the genome. And so it can identify bacteria to a genus level, but again, we're missing eukaryotes and viruses, and this technique really suffers from very poor reproducibility and a lot of false positives. Whole shotgun or metagenomic sequencing of the microbial DNA is another step forward. It's much better. It can get to strain level. It can identify DNA viruses and other eukaryotes, and then it can identify the potential metabolic pathways that are present. But what we do at Viome is another step forward. I would say it's a quantum leap forward in our understanding of the microbiome, and that's metatranscriptomic sequencing. So looking at all of the RNA, that allows us to identify all organisms to strain level, including RNA viruses, which are really proving to be a very important part of the microbiome and our overall health. We see only living organisms, and more importantly, we see what these organisms are doing. So identifying RNA sequencing, the microbe with RNA sequencing, is it allows us to see what they're doing. So it gives us a clear picture of what the microbes are consuming, what they're producing, and how they're interacting with each other. And the diagram on the right just shows how two different diets can completely change the genetic expression of the same bacteria, where that would show up as the same DNA. So this is the technology that we're using at Viome. It's a direct-to-consumer model. We can look at our customers' microbiomes and make personalized recommendations based on what those microbes are doing. This is a technology that's taken a long time to develop, but we now have access to it, and it allows us to offer this type of testing very inexpensively and very precisely to get this to a large number of consumers so they can track their internal ecosystem over time. And what we're also adding is looking at using the same technology that we're looking at the microbiome, the transcriptomic technology. We're also able to do that from a finger prick of human blood. And then these final functions, looking at the metabolites in both urine and blood, is what we're adding to the product to add a very complete picture of what's going on. So with this RNA and metabolite data, we can identify the complex pathways, the reactions that are being carried out by our microbes and by ourselves. And we can start to see what we need to do to optimize them. Now, you can imagine this produces a lot of amazing data. And that with this amazing amount of data, it's clear that our human brain, even our human microbial brain, if you want to think of it that way, just can't keep track of everything. So we need to use the power of artificial intelligence to help us understand what's going on in each person. So AI uses a knowledge-based reasoning and machine learning to produce personalized wellness recommendations delivered through an interactive chatbot style app for our customers. And if you want to get more information on this, you can go to viome.com or email businesspartners@viome.com. And I'll leave, some of the insights, some of the specific recommendations that we make if people are interested in the question and answer. And I think there we go.
Thank you so much, Helen. That was a fantastic talk. Thank you. Wow, it's pretty incredible what is inside of us and how Viome is taking and looking at genetics and applying it to food science and how food has an impact on us.